Chapter 9, Energy Metabolism, Screencast 3. In the previous screencast, we basically introduced the oxidation of glucose and the generation of ATP. We introduced all of the pathways that are necessary. We're now going to move on to how the body actually gets energy from lipid. So first off, you have to break the lipid down first, and that's going to be called lipolysis. So generally what's happening here, remember, we consume lipid primarily as triglycerides, and so that's what lipolysis actually is. It's the breakdown of triglycerides into free fatty acids and glycerol. So remember, triglycerides are very, very large molecules, so we consider them to be energy dense. Remember, when we're talking about a gram of lipid, it's actually worth 9 kcals per gram, as opposed to 4 kcals per gram for carbohydrate and protein. Then we have fatty acid oxidation. This is going to be a further breakdown of fatty acids for the production of energy, and these fatty acids will donate electrons to oxygen. This is a process that actually requires oxygen and takes place within the mitochondria. Now, triglycerides will be broken down from either those that are consumed or stored within our body and adipose tissue, and basically this is where it'll be stored after high-fat meals. During fasting, triglycerides are broken down into fatty acids. There is something known as hormone sensitivity lipase. The activity is increased by glucagon, growth hormone, and epinephrine, but decreased by insulin. So fatty acids will be taken up by cells, and they are actually shuttled into the mitochondria from the cytosol by carnitine. So here we refer to that carnitine shuttle, and remember, fatty acids are coming from lipolysis, the breakdown of triglycerides. Carnitine is actually used as a shuttle to move fatty acids into the mitochondria, where they will undergo beta oxidation, and the product of beta oxidation will be acetyl-CoA. Now, our textbook puts a very significant emphasis on acetyl-CoA, and we'll be addressing that along the way. Now, going back to the introduction of triglycerides and fatty acids, they are always made up of an even number of carbons. So to transfer energy from fatty acids, 2 carbon groups must be cleaved off, and this is used to create acetyl-CoA. And this is entering the citric acid cycle. Along the way, NADH plus hydrogen ion, FADH2, will be formed, resulting in more electron transport molecules and chemical energy storage. This is referred to as fatty oxidation or beta oxidation. And remember, fatty acids contain many, many more carbons than glucose. So there will be more acetyl-CoA groups that are formed to eventually enter the citric acid cycle. And this is why fat is actually yielding such more significant energy than carbohydrates. Once again, 9 kcals versus 4 kcals per gram. Now here we're looking at an overview of beta oxidation, and notice what's happening. We're starting off with a 16-carbon fatty acid, and notice we basically cleave 2 of those carbons. Here's our acetyl-CoA, but also notice we're going to end up with some electron transport molecules as each 2 carbons are cleaved off. Then if we look at now a 14-carbon fatty acid, we have 2 more carbon fragments that are broken off and combined with CoA to form acetyl-CoA, along with the electron transport molecules. And beta oxidation reactions will continue breaking off 2 carbons at a time and combining with CoA. So basically, with this 16-carbon fatty acid, we have 8 acetyl-CoA, 8 FADH2, and 8 NADH plus corresponding hydrogen ion. So basically, we've created a lot of acetyl-CoA, 8. So that basically will mean 8 spins of the citric acid cycle. Remember, for FADH2, eventually we will get 2 ATP for FADH2. That's 16. And we actually get 3 for each NADH and hydrogen ion. So that gives us 24. So from a single fatty acid, we get lots and lots of energy, as illustrated in this example. Now, the citric acid cycle also produces compounds that enter biosynthetic pathways. This results in a slowing of the cycle, and so not enough oxoacetate is produced. Now, cells can synthesize oxoacetate to compensate. One source is pyruvate. Now, Remember, carbohydrates are needed to create pyruvate as fatty acids create acetyl-CoA. Now, here we basically see the role of carbohydrate in beta oxidation. We start off with glucose being converted into phosphoenolpyruvate, and then we have pyruvate, which can actually be converted into oxoacetate. Here we see the acetyl-CoA molecule from fatty acids that have undergone beta oxidation, and they enter here. So notice what's happening. From pyruvate, we basically see being converted into oxoacetate. Here we have the acetyl-CoA actually going into the citric acid cycle, where it combines with oxoacetate to form citrate. Now, ketones are going to be the byproduct of fat metabolism. Now, ketone bodies will form when there is inadequate insulin, and that insulin production does not balance glucagon production. If carbohydrate consumption is too low to generate oxoacetate, then the citric acid cycle decreases. Beta oxidation continues, and this results in a buildup of acetyl-CoA, and this is what actually results in the production of ketones. Now, most ketones can be converted back to acetyl-CoA in other cells of the body. Now, the buildup of one ketone body, acetone, leaves the body via lungs, resulting in fruity, smelly breath. I use the example of juicy fruit chewing gum that used to be popular, not often popular anymore. Now here, if we look at what the key steps in ketosis are, the first, there's insufficient insulin production. Here we have a diabetic. Large amounts of fatty acids are going to be released by adipose cells. These fatty acids flood into the liver and are broken down into acetyl-CoA. Now, if we look at this high production of acetyl-CoA from beta oxidation, it actually slows down the citric acid cycle. So now we have this high amount of acetyl-CoA, and they unite in pairs to form ketone bodies. In the type 1 diabetic, too little insulin is produced, and this results in fat and carbohydrate metabolism abnormalities. If insulin is not present, cells cannot use glucose. This results in rapid lipolysis. Ketone bodies are produced in excess. This spills into urine with sodium and potassium, potentially resulting in an ionic imbalance. Now the blood becomes acidic because of the acid group on the ketone bodies, and this overall is a condition known as diabetic ketoacidosis. Now, also, we might build ketone bodies in semi-starvation or fasting. So during semi-starvation or fasting, glucose and insulin levels fall during semi-starvation or fasting. Fatty acids flow into the bloodstream and form ketone bodies in the liver. Now, heart, muscle, and kidneys, and eventually the brain will get to the point where they can actually use ketone bodies for fuel. This is an adaptive response to spare proteins from being used as a fuel source. We really do not want to use proteins for energy. That's the last thing we want to use protein for. As remember, protein has so many other functions within the body.